Hi, everybody. My name is Haritha. For those of you who don't know me, I'm a fifth-year graduate student in the Brembilla lab, and today I'm going to be talking to you about a portion of my project which investigates TNFR2 signaling and its role in oligodendrocyte precursor cells during neuroinflammation following CNS disease. So just as a brief outline for my talk, I'm going to first go into the background very briefly on the different facets for my project before delving into my actual data, showing the different functions of TNF receptor 2 signaling in OPCs. So as you all know, the Brembilla lab focuses on the disease called multiple sclerosis, which is a chronic neurodegenerative disorder that affects more than 2.5 million people worldwide. It is a T-cell mediated autoimmune demyelinating disorder in which peripheral immune T cells infiltrate into the CNS, bind to myelin fragments on antigen-presenting cells, which results in classical hallmarks of the disease, which are acute inflammation, oligodendrocyte death, demyelination, axonal degeneration, and neuronal loss. So these hallmarks will result in several different symptoms in patients, depending on the patient. So the most common symptoms include numbness and tingling, fatigue, walking difficulty, vision problems, and so on. So this MS can affect a variety of different functions, and it is a very broad and also patient-specific disease in the sense that each patient will have differing symptoms. So two of the main challenges for developing therapies for MS is, as I was mentioning, there's two challenges in developing therapies for MS. The first is neuroinflammatory damage control. So we have to be able to suppress the neuroinflammation that occurs in these patients. And in addition to that, we have to promote remyelination and repair of the CNS to actually repair the injury sites that occur. So current disease-modifying therapies are able to suppress the neuroinflammation that occurs, and so they're fairly effective in slowing down the disease and suppressing the inflammatory attacks, but they are also associated with severe side effects and they do not promote the repair of the CNS. So the lack of effective therapies is partially due to a lack of understanding of the underlying pathophysiology of MS. So our lab focuses on tumor necrosis factor, which is also known as TNF, and that's an immunomodulatory cytokine that's known to regulate inflammation, cancer, autoimmunity, and infections. So there's two types of TNF. There is the transmembrane form of TNF, which can be cleaved into the soluble form of TNF. And these two forms of TNF can bind to two different receptors. There's TNF receptor 1 and TNF receptor 2. Due to differing binding affinities, soluble TNF preferentially binds to TNFR1, and transmembrane TNF preferentially binds to TNFR2. And depending on which receptor is bound, there will be differing downstream signaling effects, which I'll go into later in this talk. So specifically in MS, it's been found that MS patients have elevated TNF levels in the CSF and cerebrospinal fluid compared to unaffected individuals. And the levels of TNF positively correlate with increased disability, as well as the rate of progression of the disease. In addition, TNF has been found in lesions and can also be correlated with the increased severity of lesions and disease progression. So in order to further understand how TNF could be playing a role in MS pathology, researchers decided to turn to animal models of MS. The most common one is known as experimental autoimmune encephalomyelitis, or the EAE model. And in this model, you can induce a BBB breakdown and immune response to myelin-specific antigens, which results in infiltration of peripheral immune cells into the CNS. And this results in an MS-like phenotype that can be scored on a scale from 0 to 6. 0 being a healthy mouse, and 6 being a mouse that died from the disease. So using this animal model, and as well as genetic knockouts, in the past several decades, there have been several conclusions or studies that have investigated the role of TNF in EAE. So we have shown before that total TNF ablation results in exacerbated EAE, suggesting that TNF does not necessarily only have a detrimental role, but also can be protective as well. And then if we look at the two different receptors, TNFR1 ablation resulted in mice that were resistant to EAE, suggesting that TNFR1 is detrimental to EAE progression. And TNFR2 ablation, on the other hand, resulted in exacerbated EAE, suggesting that the presence of TNFR2 is necessary to suppress the clinical scores during EAE. Our own lab has used a pharmacological approach and inhibited soluble TNF signaling via TNFR1 and found that by leaving TNFR2 signaling via TNFR2 still active in the mouse, it results in improved long-term recovery and remyelination, suggesting that soluble TNFR1 or TNF signaling via TNFR1 is detrimental, while transmembrane TNF signaling via TNFR2 is protective. So, and there have been various studies that show that TNFR2 signaling in particular can promote cell survival, anti-inflammation, and remyelination, while TNFR1 signaling usually results in apoptosis and inflammation. So our lab is interested in studying the effect of, or studying how exactly TNFR2 controls or suppresses the inflammatory response necessary for EAE development, and how does TNFR2 control or promote the remyelination capacity necessary for repair following injury. In order to answer those two questions, our lab has decided to use a genetic approach where we selectively ablate TNFR2 in this it is present in the CNS. So we found that TNFR2 is present in astrocytes, oligodendrocytes, and precursor cells, as well as microglia. So Han Gao, a previous grad student in our lab, investigated the role of TNF2 in microglia. Placido Leon, a current postdoc in the lab, is looking at the role of TNFR2 signaling in astrocytes. And the basis for my project is from work that was done by Pernille Madsen, a previous grad student, looking at the role of TNFR2 signaling in the whole oligodendrocyte lineage. So she generated these CMP-CRE TNFR2 floxed mice in which the TNFR2 is ablated selectively in the whole oligodendrocyte lineage. And she found that when EAE was induced, see here that the conditional mouse in the have scores compared to the controls in. And this suggests that the presence of TNFR2 signaling in oligodendrocytes is protective during EAE. And you can see here that the main difference in scores is at the chronic and at the acute phase. And each of these phases is results in a different outcome, or the clinical scores in each of these phases is attributed to a different underlying mechanism. So usually during the acute phase, these clinical scores are due to inflammatory attacks that are occurring due to the infiltration of immune cells, while during the chronic phase, these scores are attributed to mechanisms of remyelination that are or are not occurring in these mice. So I'm not going to go into all of the data that Pernille generated, but just in brief, she was able to conclude that TNFR2 signaling in the whole oligodendrocyte lineage is necessary for oligodendrocyte differentiation, and it suppresses the inflammatory response after EAE induction. Now, one of the pitfalls, I guess you could say, for her animal model is that she is ablating TNFR2 in the whole oligodendrocyte lineage, which means it is being ablated in the OPCs, pre-oligodendrocytes, mature oligodendrocytes, and the myelinating ones. So it's difficult to parse out exactly which cell type may be influencing EAE progression, as well as how each of these different cell types could be playing a role. So in vitro evidence from her work suggested that OPCs may be a major contributor to TNFR2 dependent regulation of EAE. So, which was the basis for the beginning of my project, which is to look into how does TNFR2 control OPC response to an inflammatory environment like in EAE. So in order to answer this question, we decided to first start off with in vitro assays. So I generated primary OPC cultures from wild type and TNFR2 knockout mice using the Miltenyi magnetic labeling system. And so this is how these OPCs look when you culture them. Olig2 is a marker for the whole oligodendrocyte lineage, and PDGFR alpha is a marker for OPCs. And so after culturing these OPCs for 3 days, they were either not stimulated or stimulated for 3 hours with TNF, interferon gamma, and IL-1 beta. These are 3 cytokines that are known to be highly expressed in EAE as well as in MS patients. So this is to stimulate or to mimic an inflammatory environment that is similar to what happens in vivo. And after the 3-hour stimulation, the cells were collected either for RNA, protein, or histology. And when I did a qPCR on these samples, you can see here that following the 3-hour stimulation in the wild type cells, we see almost a 7-fold increase in TNFR2 expression. And obviously, as expected, this increase does not occur in the TNFR2 knockout cells. This increase in TNFR2 expression, as well as the reduction in TNFR2 in the knockouts, is also correlated with downstream signaling. So AKT, the AKT signaling pathway is known to be downstream of TNFR2. And you can see here that the phosphorylation of AKT is significantly reduced in the knockouts, showing that in our germline knockouts, there not only is TNFR2 ablated, but the downstream signaling cascades are also impaired as well. So this led us to want to further investigate this, and we asked the question, what is the role of TNFR2 signaling in OPCs when exposed to an inflammatory environment? So in order to get a more broad view of exactly what is intrinsically different in OPCs in response to inflammation, as well as TNFR2 expression, we decided to conduct an RNA sequencing experiment in which we did the exact same stimulation and then sent off the samples, sent off the RNA to the Beijing Genomics Institute, which gave us back 30 million paired and 100 base pair reads that we aligned to the mouse genome and conducted differential gene expression analysis. So just to reiterate, I was able to generate 4 groups. So there's the wild type cells that were non-stimulated, the wild type stimulated, and then the knockout non-stimulated and the knockout stimulated. So I have a total of 3 replicates per group, so a total of 12 samples. And when I conducted the differential gene expression analysis for each of these comparisons, I found that hundreds of genes were differentially expressed with a fold change greater than 1.5. So it can be very overwhelming to go through this kind of data because it's so much and it's hard to parse out. So in order to make it a little easier on myself with the data mining, I decided to separate out each of these groups and ask exactly what are the genes that are selectively altered within each comparison. So in order to do this, I created this graph that's called an upset plot. It's sort of like a Venn diagram, so I'll walk you through it. On the y-axis, we have the intersection size. So this is the number of genes within each specific group. And then down here are each of the different comparisons. So for example, if we take the circle here that's in blue, So these are the genes that are only altered or only upregulated in the TNFR2 knockouts, non-stimulated versus the wild-type non-stimulated group. And so there are 347 genes that are selectively altered between the two genotypes under basal conditions. So if we look at each of the comparisons here that I have colored, there's several gene ontology biological processes that are altered or upregulated following stimulation or due to TNFR2 knockout in these cells. So I want to specifically highlight here, we see that there's metabolic processes as well as inflammatory response that shows up multiple times within these comparisons. So these were two pathways of interest that we wanted to follow up on. And similarly, if we look at the downregulated genes, we found that the cell cycle and cell division, as well as lipid metabolism, were biological processes that were downregulated following stimulation in the knockouts compared to the wild types. So from this gene ontology enrichment analysis, we identified three pathways of interest: the immune system response, cell cycle, cell division, and metabolic processes. So if we first go into the immune system response, I pulled out several genes that are known to regulate immune cell proliferation or migration. And you can see here that following stimulation, both wild type and TNFR2 knockout cells upregulate several of these chemokines and cytokines that are known to promote immune cell proliferation and migration. There are even several genes that are actually TNFR2 dependent. So CXCL10, CCL2, CXCL12, for example, they all are upregulated in both the wild types and the TNFR2 knockouts, but this upregulation is further exacerbated when TNFR2 is ablated, suggesting that TNFR2 could play a role in OPCs, could play a role in microglia or immune cell proliferation. In order to test this, we decided to, or well, first, we conducted a mesoscale analysis to see if those RNA changes correlate with protein differences. And as you can see here, following stimulation, the OPCs drastically upregulate these cytokines and chemokines, and some of them are even TNFR2 dependent, like interferon gamma and IL-10. In order to test this out functionally, we decided to conduct a microglia proliferation assay in which I collected conditioned media from these stimulated OPCs and then applied them to the CIMA9 immortalized microglia cell line overnight and conducted either a phagocytosis assay or a proliferation assay to see if these OPCs that were stimulated release factors that could actually release factors into the media that could influence microglia function in vitro. So first, for the phagocytosis assay, here's just some representative images here of how these cells look. In blue, I have DAPI, in green is the beads, and red is IBA1, which is a marker for microglia. And as you can see, when wild type or TNFR2 knockout non-stimulated conditioned media is added, there is fairly low levels of phagocytosis of these green fluorescent beads. However, when stimulated conditioned media from stimulated cells is added to the microglia, we see this qualitative increase in the phagocytosis of these beads. This is fairly new data, so I haven't completed the quantification yet, which is why I'm not showing you that data, but it would be interesting to see whether or not this increase in phagocytosis of the beads is TNFR2 dependent or not. When we look at microglia proliferation through EDU incorporation into these cells, we can see that the cells under basal conditions, or when they're have non-stimulated conditioned media added to them, there's very low levels of proliferation. However, when the stimulated media is added to these cells, we see increased EDU immunostaining in these microglia, suggesting that the OPCs are indeed releasing factors in response to that inflammatory stimulation that regulate microglia proliferation. Again, I'm still in the process of quantifying all of this, so I don't know if this process is TNFR2 dependent or not yet. So just as a brief summary, what I've shown you so far is that OPCs, even wild-type OPCs, can produce cytokines and chemokines when exposed to a neuroinflammatory environment. And some of these factors are upregulated in a TNFR2 dependent manner. And in response to stimulation, OPCs are indeed capable of actually releasing these factors that, at least qualitatively, you can see promote microglia proliferation and phagocytosis in vitro. So some future experiments for this part of the project is to look at the quantification of the microglia phagocytosis and proliferation, as I mentioned before. And in addition, I want to conduct a multiplex analysis of the media that the cells were cultured in to see what inflammatory mediators are secreted by OPCs. And that should give us a better idea of exactly what factors are TNFR2 dependent or not. So moving on to the next pathway of interest here, we have the next one is cell cycle and cell division. So I specifically looked at genes relating to OPC proliferation and differentiation. So you can see here that overall it seems that after stimulation, there's this downregulation of OPC marker genes, as well as downregulation of positive regulators of oligodendrocyte differentiation, and interestingly, also downregulation of negative regulators of oligodendrocyte differentiation after stimulation. So this suggested that that overall, it seems that following stimulation, these genes that are involved in cell division or cell differentiation just seem to be downregulated in general. And in order to see if that results in any functional differences in these cells, I decided to conduct a differentiation assay in which wild-type or TNFR2 OPCs were stimulated and then cultured for 4 days with T3 and CNTF, which are oligodendrocyte pro-differentiation factors that will promote the differentiation into oligodendrocytes. And when we quantify the number of O4 positive cells, so these are immature oligodendrocytes, in our different conditions, we observe this slight reduction in the number of O4 positive oligodendrocytes in the basal TNFR2 knockout condition compared to the non-stimulated wild types, suggesting that in basal conditions, this initial differentiation into O4 positive cells could be impaired. If we look at a later stage of differentiation through MBP expression, which is expressed by oligodendrocytes that are capable of producing myelin, we see that overall it seems that both wild types and knockout cells that are stimulated have a reduced capacity of producing MBP or differentiating into MBP positive mature oligodendrocytes. And so this is just a representative immunostaining of these cells. If we take a closer look, you can see here that the wild type non-stimulated cells form these very nice nets of myelin in the plate. On the other hand, if we look at the TNFR2 knockout cells in non-stimulated conditions, while these cells still seem to express MBP, they have a very different morphology to them. It's much more ramified, and even the few cells that do form these nets outward have this very broken kind of structure to them. On the other hand, if we look following stimulation, wild-type cells do not seem to express much MBP and instead have this very compact, ramified structure, while the TNFR2 knockout cells following stimulation also reduce their MBP expression, but seem to not be as compact as the wild type cells. So this suggests that even though we don't see much differences in the actual number of cells expressing these different markers, it's possible that these, instead there's morphological differences that are TNFR2 dependent, both under basal and stimulated conditions. So in summary here, I'm just showing you, or I've shown you so far that OPC proliferation and differentiation associated genes are mainly downregulated following stimulation, some in a TNFR2 dependent manner, some not. And this downregulation suggests that there's this impairment in cell cycle or cell differentiation, and we see that following stimulation, these cells have reduced differentiation into MBP positive cells, and specifically OPC differentiation is impaired in a TNFR2 dependent manner under basal conditions compared to wild type OPCs. So some future experiments here. I definitely want to conduct a morphological analysis of these oligodendrocytes in vitro with and without stimulation. And it would be interesting to do a qPCR of RNA extracted from these differentiated oligodendrocytes to see if there's any TNFR2 dependent differences in the myelin-associated genes in this assay. So moving on to the metabolic processes. As we all know, metabolic pathways are highly complex. Even a slight change in cell metabolism can result in cell stress and dysregulation of cell activity and cell function. So when I looked further into different metabolic genes, I came across, I specifically looked first at the TCA cycle, which we know is important for energy metabolism. And so this is just a pathway analysis that shows here the different genes that are differentially regulated within the TCA cycle between each comparison. So on the left here, we have, it's comparing the TNFR2 knockout non-stimulated versus the wild type non-stimulated group. And as you can see here, there are quite a few genes that are upregulated in the TNFR2 knockouts compared to the wild types, suggesting that even in basal conditions, energy metabolism is dysregulated in a TNFR2 dependent manner. Now, following stimulation, there's still several genes that are upregulated in the TNFR2 knockouts compared to the wild types, but not as many as the basal conditions. But this makes sense because in following stimulation, the wild type cells are also undergoing cell stress. So the fact that we still see several genes that are upregulated in the TNFR2 dependent manner just shows that TNFR2 does regulate energy metabolism and even in inflammatory conditions. So one specific pathway that I found to be interesting after doing further reading is through the NRF2 signaling pathway. So here I'm just showing the NOS2, which is encodes for nitric oxide synthase. So it promotes the production of free radicals that can increase cell stress and reactive oxygen species. And you can see here that in the TNFR2 knockouts, we have the significant increase in this gene. In addition to that, we see increased expression of NRF2, which is an antioxidant gene. So, and you can see that the TNFR2 knockout cells have increased expression of NRF2 as well. And this increased expression of NRF2 suggests that the TNFR2 knockout cells are undergoing possibly higher levels of stress than the wild types following stimulation. As a result, they're upregulating these antioxidant genes. So a little bit of background on this NRF2 signaling pathway. Under basal conditions, NRF2 is usually bound by KIP1, and that results in proteasomal degradation. However, following oxidative stress, NRF2 dissociates from KIP1, translocates to the nucleus, and promotes the production of antioxidant response genes that play a role in many different metabolic processes, ranging from lipid metabolism to maintenance of cell homeostasis, apoptosis, NADPH generation, and so on. So it has pretty broad effects on controlling the metabolic stress and energy metabolism that occurs in the cells. So if we look at specific genes that are downstream of NRF2, I've noticed that there's this upregulation of several of these genes in the knockouts compared to the wild types, suggesting that this pathway indeed is regulated by TNFR2. So just in summary here, what I've basically shown so far for this portion is that TNFR2 ablation in basal OPCs seems to result in increased metabolic stress. And following after stimulation, TNFR2 ablation seems to further exacerbate this stress. And several of these genes that are differentially expressed are downstream of NRF2. So in order to determine if this is actually relevant or not, I'm going to be conducting a Western blot to see if NRF2 translocation to the nucleus in these different conditions is TNFR2 dependent or not. And I'll also be testing KIP1 inhibitors in vitro to see if the gene expression changes observed in the TNFR2 ablated OPCs can be reversed. So just as a brief summary of what we see so far in the in vitro data, under basal conditions, wild-type OPCs have two functions. They can either proliferate or differentiate into mature oligodendrocytes. In a neuroinflammatory environment, wild-type OPCs seem to reduce their differentiation or reduce their cell cycle genes, which results in reduced differentiation and possibly proliferation. And in contrast, they upregulate cytokines and chemokines that will contribute to the inflammatory environment of these cells. So on the other hand, TNFR2 knockout OPCs seem to already be undergoing some sort of cell stress, and this results in slightly reduced differentiation into mature oligodendrocytes. And following stimulation, these knockout cells have even more exacerbated cell stress than the wild type OPCs, and this results in much higher levels of increased expression of these chemokines and cytokines that will contribute to the inflammatory environment. So obviously that was all in vitro data, so we wanted to make sure or to confirm whether or not these changes we see occur in vivo as well. So in order to test this, we generated a mouse model called the PDGFR alpha CRE, TNFR2 flox, EYFP mice. So in these mice, the CRE recombinase is under the transcriptional control of PDGFR alpha, which is a promoter that's expressed in OPCs. And when these mice are administered with tamoxifen as adults, they will conditionally ablate TNFR2 and simultaneously express EYFP in the OPCs, specifically in the CNS. The corresponding controls for these experiments are the PDGFR alpha CRE EYFP mice. So in this case, the TNFR2 is not floxed, so the TNFR2 is still present, but upon administration of tamoxifen, we will still get expression of EYFP in the recombined cells. So just as a brief summary, I've already characterized the recombination is about 75% or 70% in the PDGFR alpha positive cells that also express EYFP. And there's about a 50% ablation of TNFR2 in these recombined cells. And just as a disclaimer, I do want to mention that about about 1% of these PDGFR alpha cells. In the spinal cord, I found to also express PDGFR beta, which is a marker for fibroblasts. So there is this minor population in which in the knockouts, TNFR2 might be ablated. In addition, I also did some basal behavioral analysis under naive conditions and found that there's no changes observed in locomotor behavior through the open field and coordination via rotarod. So again, I want to bring back the EAE model here because we are interested in seeing whether or not TNFR2 ablation in OPCs will result in will alter the clinical progression of EAE. So just as a reminder, when EAE is induced, this results in an MS that could be scored on a scale from 0 to 6. So when I induce this EAE in this mice, on the y-axis here, we have the score. A score of 3 is considered to be full hind limb paralysis, just so you have an idea of what these scores mean. And as you can see here, the knockouts in the black circles seem to have this earlier onset and earlier peak disease following EAE induction. And I mentioned this very briefly before, but this acute phase, or the clinical scores during this acute phase, are mainly attributed to the immune cells that are infiltrating from the periphery and attacking the CNS. So the fact that we see this earlier onset and earlier peak disease suggests that TNFR2 signaling in OPCs does regulate the initial neuroinflammatory response following EAE induction. So in order to see if that's actually the case, I decided to conduct a flow cytometry experiment in the spinal cord at pre-symptomatic, acute, and subacute time points, and I stained for different immune cells, T cells, B cells, neutrophils, and so on, to see if the actual infiltration of these cells is altered in a TNFR2 dependent manner. And you can see here that we see this accelerated CD4 positive T cell infiltration into the CNS, but CD8 T cell infiltration is not altered. In addition, we see increased natural killer cells in the CNS at the acute time point, as well as increased neutrophils in the acute time point as in the TNFR2 knockouts compared to the controls. We also see a shift in the activation of macrophages. So there's really no difference in the actual number of macrophages, but we see this shift in MHC2 expression, with the knockouts expressing more MHC class 2 than the controls at this acute stage. In addition, we also see an increased number of microglia in the knockouts compared to the controls, as well as increased activation of these microglia. So if we go even earlier, so even as early as 8 DPI, at this stage, none of the mice are sick, and they're still several days before they actually show symptoms. We can qualitatively see this increase in IBO1 immunostaining in the spinal cord of the conditional knockout mice compared to the controls, suggesting that this TNFR2 ablation in these OPCs seems to promote or regulate microglia even in pre-symptomatic EAE and early on in the disease. So just as a summary here, I've shown that TNFR2 ablation in OPCs during the acute stage results in accelerated infiltration of CD4 T cells, neutrophils, as well as increased activation of macrophages and increased microglia number and activation, even as early as 8 DPI, or 8 days post-immunization. So this overall suggests that TNFR2 signaling in OPCs suppresses neuroinflammation during EAE, which it can also be correlated or matched with the in vitro data that I showed earlier. And there's still a lot to be done for this project. So I just showed you some representative images of the IBO1 staining at EAE 8 DPI. I will be doing a serological quantification of these cells to make sure to determine if that difference is statistically significant. In addition, I'm in the process of fact sorting out the YFP positive cells from the pre-symptomatic EAE stage to determine what genes are differentially regulated in a TNFR2 dependent manner between the knockouts and the controls, and compare that to the RNA sequencing data I showed before. I'm specifically interested in looking at the NRF2 signaling pathway genes. And then in addition to that, I'll be doing a multiplex analysis to look at the exact inflammatory mediators that could be playing a role in this earlier onset and earlier peak disease, as well as doing a histological analysis of demyelination at the various stages to see if the demyelination or remyelination is regulated by TNFR2 signaling OPCs as well. So finally, I'd like to acknowledge everyone in our lab for all of their help, as well as our collaborators in Denmark. The Lambertson lab helps out with all of the mesoscale analysis and data. The Bach lab, who have provided us with one inhibitors that I'll be testing, as well as my thesis committee for all of their input into this project. Jan from the Miami Project Imaging Core for all of her help with the stereology and the celomics. Oliver from the flow cytometry core, who's been absolutely essential for all of the flow cytometry and cell sorting experiments. Matt, who was a previous grad student in the Lembix lab, who taught me the very basics of RNA sequencing analysis and data, and obviously BGI for actually doing the RNA-seq and generating the data, and all of you for listening to this talk. And I'll be happy to take any questions now. There aren't any.
All right. It seems, does anyone have any questions? You can either go ahead and grab the mic. I will clear the mic so that you can unmute yourselves, or you can go ahead and ask a question via chat.
Okay.
Can you hear me?
Yes. Yeah.
Oh, good. Excellent talk. Thank you very, very much. Just really exciting work that you're doing in the laboratory of Dr. Brambilla. Congratulations on all the outstanding data. What do you think about translating some of your findings ultimately into the clinic to target MS and other clinical problems? You're showing very nice cellular mechanisms in terms of inflammation and things like that, but how would you go about thinking about the next stage in a more translational study, if you will? And again, thank you very much for the lecture.
Yeah. So I haven't mentioned this in this talk, but the whole, the overall goal for our lab is to basically not only investigate the underlying mechanism of TNFR2 in all of these cell types, but also figure out a way to harness this protective function of TNFR2 and maybe be able to translate that into therapies or identify compounds that could enhance this TNFR2 signaling, which would eventually result in a protective effect in these cells, or in MS, in theory. So that is the overall goal for the lab. So we are doing several preclinical studies to see if that would be possible. We have overexpression models that we are currently investigating to see if enhancing this TNFR2 is protective, not just showing that knocking it out is detrimental, but it's important to see that enhancing it would be protective. So that's something that is currently ongoing in the lab. Yeah.
You're using one particular model of MS. Are the other models that ultimately may be needed if you found a compound that you would want to take to the clinic, do you think? Is that being discussed in the literature at all? Is there a non-human primate model of MS, for example, that people use, or what?
There is a primate model of MS, and I am not too familiar with that, in all honesty, but definitely that would be something. It's similar to EAE, I believe.
Okay. Right.
So.
That's what I think as well. Thank you very much. Appreciate it.
Mm-hmm.